Welcome to Click for Biology. We're looking at plant nutrition and we're going to explore another plant experimental technique where we test a leaf to show that it has made starch. The particular technique that we're going to look at also includes a process called destarching. So let me try to explain the context of this before we look at the actual technique. We know that in photosynthesis, a plant will produce glucose, and we also know that glucose is stored in plants as a polymer called starch, which is a chain of glucose molecules. When the plant requires glucose for respiration, it will then break down the starch back to glucose and respire that glucose. If we are going to demonstrate that a plant is making starch, what we're actually saying is that the plant is doing photosynthesis, that they are making glucose, and that glucose can be detected by detecting starch. However, if we test a leaf for starch, how do we know that that starch wasn't made before your experiment began? This is the basis of destarching. We take a plant and we know that in the plant leaf there will be starch. That starch, of course, is made up of glucose. And so the first thing that we do before we do any experiment is to take the plant and place it in the dark for up to 36 hours. In the dark, the plant will take the starch and break it down to glucose. The plant has to do this because, of course, in the dark, there is no photosynthesis. So in the dark, the plant takes its starch and breaks it down to glucose, and then the glucose is broken down by respiration. Therefore, after a 36-hour period of being in the dark, the plant no longer has any starch. We could prove that by carrying out a starch test on this particular leaf. Now, to demonstrate that plants need light for photosynthesis, we would take the destarched plant and then place it in light for 24 to 36 hours, and then we would carry out the test for starch on the plant which has been destarched initially, but then placed back in the light to make fresh glucose, which will be stored in the leaf as starch. Let's look at how you test the leaf either at B or at C for the presence of starch. The first issue is that on the upper surface of the leaf, there is a waxy cuticle. If you pour iodine solution onto the leaf, the iodine will simply run straight off. So we need to remove the waxy cuticle by boiling the leaf or placing the leaf in boiling water, and this will melt the actual cuticle. Having done that, we have a leaf now that no longer has the cuticle. The problem that we now have is that the chlorophyll is so strong in its green color that we cannot see the color change of the iodine solution that we will use at the end. So now we need to remove the chlorophyll, and we do that by warming the leaf in ethanol. Ethanol will dissolve the chlorophyll and bleach the chlorophyll out, leaving behind a dechlorophyled leaf which will no longer have the green color, but will probably have a pale white color. It's important to realize that when we do this step, that we do not use Bunsen burner flames or any source of ignition, as ethanol is highly flammable. Instead, one should use a water bath. Having boiled away the waxy cuticle and having removed the chlorophyll, we have one step left, which is to rinse the leaf with water so that we're removing the ethanol from the surface of the leaf. Then we apply drops of iodine solution to the leaf surface. It's sometimes necessary to gently touch the leaf surface with a pair of forceps to break up the leaf structure. And then what we should see is that the brown iodine solution will turn to a blue-black color, showing that there is starch present. This is the way which we demonstrate the presence of starch in a leaf to show that it has been made in photosynthesis. Okay, let's just check our understanding. Let's do a 3-minute quiz. Pause the video, answer these questions, and then check your answers in the description down below the video. And when you've done that, you can come down to here and follow the link to the next video in the playlist. The playlist is linked as well. The channel link is there to subscribe, and a thumbs up would be very pleasing as well. Thank you for listening.